So today in our LIBREhub project seminar series, we welcome Eleonora Perego. How do you pronounce it?
Perego, yeah, quite good.
And Eli Slenders.
Perfect.
Are two postdocs that recently published a very interesting article of an open source hardware instrument to democratize single-photon microscopy, which I think is interesting to a lot of people in our project. Just briefly again, as a reminder, so the LIBREhub project is aimed at helping people around the world, but focused on Latin America in particular, to access interesting advanced research tools by distributing knowledge and training in hands-on workshops around building their own open source hardware instrumentation, especially low cost and open source hardware, so that people can access techniques that otherwise could not be affordable or maintainable across Latin America. And so in that context, I'm also very excited to hear today's seminar on some advanced detectors. And I saw that you developed some electronics to read out the detector and process the data, probably some software as well, but that we will hear from you. So you're both two promising postdoctoral researchers. And in the advertisement, we've already foreseen that both of you will probably soon be able to get a professorship as well. So slight promotion. Thank you very much for joining us today.
Thank you for the invitation.
Yes, thank you very much. We will share the screen.
Sharing.
Should we go to the presentation one first?
Let me know if there's anything I can do to help.
That way.
Okay.
Perfect.
Okay. You see the presentation in full screen mode?
Yes.
Okay, perfect. So thank you very much. Thank you for the kind introduction, and thank you for giving us the opportunity to present here today. We're very happy, very excited to present to you the Bright-Eyes TTM, which is an open source time tagging module specifically designed for single-photon microscopy. And this is a project that started a couple of years ago with a former PhD student in our group, Alessandro Rosetta. And last year we published a paper on this Bright-Eyes TTM. So I think it's a very nice moment today to present this time tagging module. The talk today is split into two parts. In the first part, I will discuss the technical aspects of the TTM, how it works, and the key specs of the device, let's say. And then in the second part of the talk, Eleonora will discuss more of the applications of the TTM. Both Eleonora and I are working in the molecular microscopy and spectroscopy group here in IIT, which is a group led by Giuseppe Vici-Domini. And in our group, we work on the development and we use novel optical microscopy techniques. So we have several different projects running in parallel, but there is one key denominator, let's say one key thing that links all of our projects. And that's what you see on the right, which is this detector here, which is a SPAD array detector, a single photon avalanche diode array detector. And as the name suggests, it's a detector composed of multiple detector elements of multiple pixels. And this gives you extra spatial information that you can use for various applications. Yeah, but we will come to that in more detail later. Let's start first with a brief recap of a conventional confocal laser scanning microscope. So in a confocal laser scanning microscope, you have an excitation laser here depicted in blue that is reflected by the dichroic beam splitter and then focused by the objective lens onto the sample, where it creates a diffraction limited spot of light. And the sample is typically stained with fluorescent molecules, which can absorb the laser light and then emit fluorescence, which is again collected by the objective lens. And because that's different color, it can pass through the dichroic beam splitter, and then it's focused onto a detector, a single element detector, after passing through the pinhole. And the pinhole serves as a way to block light, emission light that is coming from out of focus planes. So in the end, the light that reaches the detector is only coming from a very thin region around the focal plane of the objective. And so then what you do to make an image is you use the galvanometric scan mirrors that move the laser beam in the X and Y direction. And for each position of the laser beam, then you measure the intensity of the fluorescence with the detector, and then you can generate an image. Here you see it in this animation. So you have your object that you scan with a laser beam. For every position of the laser beam, you have this single element detector that registers a certain intensity value. And then you plot this intensity as a function of the laser position, which then gives you an image of your sample. This is a conventional confocal laser scanning microscopy, which is very popular and very powerful, but still it has some limitations. And one of these limitations is that you lose information by spatially and temporally integrating the signal. What do I mean by that? Well, you have a single element detector, so it means that you don't have any information on where in the image plane the photons arrive. You only make a distinction between the photons that pass through the pinhole and the photons that are blocked by the pinhole. But all the photons that pass through the pinhole are integrated over the active area of the single element detector, regardless of their position. So you lose spatial information. At the same time, very often you also lose temporal information. And the reason is that in a laser scanning microscope, typically you integrate the signal over the pixel dwell time of your imaging system. And so you don't have information on the arrival time of every individual photon. So you also lose information on the temporal side. To solve these limitations, what you can do is image scanning microscopy. In image scanning microscopy, you replace the single element detector with an array detector composed of, for example, 5 by 5 pixels, as you can see here, and you place it in the image plane of the microscope, so where you normally have the confocal pinhole. In this case, so you don't have a pinhole, but the size of the detector itself is about 1 to 1.4 area units, so it is very similar in size compared to the conventional confocal pinhole. But so what happens now is you scan your sample, and for each position of the laser beam, you don't have a single intensity value, but you have a small image, which we call a micro image. You can see it here in this animation. So you scan the sample with your laser beam, and for each position, you record an image which only has 5 by 5 or 7 by 7 pixels. It's a small micro image, but this gives you extra spatial information. So at the end of the scan, what you have is a 4-dimensional data set. You have the intensity as a function of the 2 coordinates xs and ys of the laser beam and the 2 coordinates of the detector element xd and yd. So we have a series of micro images, but you can also think of it as I have a confocal microscope with 25 channels. And so at the end of the scan, what I have is not a single image, but I have 25 images. You can also see it better maybe in this animation. So again, you have the object, you scan it with a laser beam, and for each position, you have a micro image. But alternatively, you can also think of it as, okay, every channel of my microscope, every pixel of the detector registers an image. So at the end of the measurement, I have 25 images. These 25 images are not all exactly the same. There are slight differences between these images. And to understand that, we can look at the point spread functions coming from these different detector elements. That's what you see here on the left. So here you see for every pixel, the corresponding point spread function. And if we ignore boundary effects near the edge of the detector, then you can see that most pixels have a Gaussian point spread function, but the position of this Gaussian is shifted with respect to each other. For example, if you look at this pixel here, then the position of the point spread function is shifted slightly towards the right and in the upward direction with respect to the central element point spread function. And this is caused by the fact that the pixel is displaced with respect to the optical axis. So this is a small displacement. And this displacement you also see in the corresponding images. So all these 25 images are very similar with respect to each other, but they're all shifted with respect to each other. And so what you typically do in post-processing is...
Sorry? Can I ask this question? Could you explain in a few more sentences the point spread function? I'm just interrupting because I realize that we have quite an interdisciplinary audience, so it might not be
Yes.
clear to everybody what this is.
Yes, sure. So the point spread function of a system is the image that you get when you image a point source. So if you have in your sample a single dot, let's say, then the image you get is not a single dot anymore because of the diffraction limit, but instead you have a vague, typically Gaussian shaped blur that you see in your image. And that's what we call the point spread function. So it's the image of a point source. And this is the reason for the resolution limit that you typically have in a confocal microscope. And also here in our detector, we have 25 pixels. So with each pixel, there is a corresponding point spread function. And they all look very similar, but they're slightly shifted with respect to each other. And so the images that you take, they depend not only on the sample that you have, but also on the point spread function that you have. And since the point spread functions are all shifted with respect to each other, also the images that you observe are shifted with respect to each other. So what you then do in post-processing is you find these shifts and then you undo the shift. So you shift everything back to the right position. And then you have 25 images that are basically identical and you can simply sum them all up. And then you have one super resolved image. So an image that has a better resolution than with a normal confocal microscope. Here you see an example of experimental data of a tubulin network imaged with a confocal microscope and with ISM. On the left here, you see the image that you get when you close the pinhole of the confocal microscope. So when you close the pinhole, you block a lot of light. And because of that, the signal to noise ratio is very bad. But the resolution is actually better than when you open the pinhole. But typically you don't do that. You don't close the pinhole to such a small value just because of the fact that then you lose so many photons. And so the signal to noise ratio is really bad. Typically in a confocal microscope, you open the pinhole to about 1 to 1.4 area units in order to collect more photons. And then the signal to noise ratio is a lot better, but you lose spatial information. And in ISM, you have the best of both worlds. You have both a good signal to noise ratio because the total size of the camera is about 1.4 area units. So you have the same number of photons that you have in a confocal microscope, but every pixel of your camera is very small. And so every pixel acts as a closed pinhole with a good resolution. So when you combine these two effects, you have an image that has both a good resolution and a good signal to noise ratio. And that's the idea of ISM, image scanning microscopy. In order to implement this, you need an array detector. And so a couple of years ago, together with Polytechnic of Milan, we designed the first single photon detector array designed specifically for laser scanning microscopy. And here you can see one of these prototypes. So this is a detector composed of 25 pixels arranged in a 5 by 5 array. And each pixel is a single photon avalanche diode. So it's a detector with single photon sensitivity. And in this prototype, you can even see like all these cables. Every cable here links, let's say, or brings the signal from a single pixel to the acquisition device. So this is one of the first prototypes that we installed in a laser scanning microscope. So this SPAD array, the single photon avalanche diode array, like I said, is composed of 25 elements in a raster array pattern. The total size is about 1.4 array units. And so this allows you to sample in space with sub-diffraction precision the photons, because now you have information on where the photons arrive in your image plane. But not only that, you also have temporal information, because every pixel is a single photon avalanche diode, meaning it has single photon sensitivity, and it can register the arrival time of a photon with about 150 picoseconds precision, which is much better or much lower than the typical photophysics that you have, such as the fluorescence lifetime. Typically, fluorescence lifetime is in the order of several nanoseconds. And so if you want to collect this information, you need to have a detector that has a much better temporal precision than the fluorescence lifetime. And a SPAD array detector has temporal resolution of about 150 picoseconds. Here on the right in this table, you see some other specifications of the spot array detectors that we typically use in our daily work. So the photon detection efficiency is maximal around 500 nanometers, and then typically it decreases towards longer wavelengths. The fill factor is about 78%, meaning that you always have a bit of dead space in between the pixels. So when a photon arrives or when a photon hits this dead space, you don't detect those photons. So you try to limit this as much as possible. And one way to do that is by installing a microlens array in front of the detector. And in this way, you can increase what we call the fill factor to about 78%. The dead time of the detector is around 30 nanoseconds. So it means that a pixel is blind for 30 nanoseconds after detecting a photon. But since we have an array detector, we have many pixels, and even when one pixel is blind, let's say, then all the other pixels are still active. So this is not much of an issue in typical applications. And then there is a thing called the dark count rate, which is a property of this type of detector in which you detect counts even in the absence of light. And of course, this is something that you don't want. It's some type of noise. But typically, this is below 100 hertz per pixel. So it's much lower than the typical photon count rates that you have when you do fluorescence microscopy. So you have subdiffraction spatial sampling of your photons, and you have the temporal information to measure, for example, the photophysics of your sample. But the question is, how can you measure this temporal information? Because the temporal information, you want to register this with picosecond range precision, and you want to do this for all the 25 channels in parallel. And a couple of years ago, when we started this project at the market, there was no device that could, in an easy and user-friendly way, time tag individual photons in all 25 channels simultaneously with good precision. And so that's why we decided to build our own device called the Bright-Eyes TTM. So the Bright-Eyes TTM is an open source module for time tagging individual photons, and it's specifically designed for single photon microscopy. It's open source, so it means that everything you need, all the hardware components that you need to build, they are available online on a Read Docs page. So that you can easily find out what you need to buy in order to build it, and instructions on how to build it are also there. And also the software that we developed for running this time tagging module and for analyzing, processing the data is available on the GitHub page of our lab. So everything is open source in order to make it easier for other labs to implement their own Bright-Eyes TTM. So what is photon time tagging? Let's have a brief recap of the principle of photon time tagging. Typically, when you do photon time tagging in the context of fluorescence microscopy, is you have a laser beam that is pulsed, and you're interested in measuring the arrival time of the fluorescence photons with respect to the last laser pulse. And when you make a histogram of these arrival times, you typically get an exponential decay, and the characteristic lifetime of this decay gives you information on your sample, on the fluorophore, and so on the sample. And so what we want to do is the same, but with 25 channels, because we have 25 pixels. In reality, the situation is a little bit different, because typically you don't measure the time between the excitation and the emission, but rather you do the other way around. You wait until you observe an emission photon, and then you measure the time to the next excitation. The reason is that usually you have much fewer photon events than laser events. So you just wait until you observe a photon, and then you start measuring the time between this photon and the next laser pulse. And then you also know the time between that photon and the previous laser pulse, and you can build this histogram. So there are three types of channels that you have. On the one hand, you have all the photon channels, so 25 for a 5x5 array detector. Then there is a laser sync channel, because you want to know the arrival time or the moment in which the laser fires a pulse. And then typically you also have some reference channels which include, for example, the pixel, line, and frame clock of your measurement. For example, if you do a FLIM measurement, a fluorescence lifetime imaging measurement, then you're moving the laser beam over the sample. And so you want to know at any given moment in time where the laser beam is. So you need this extra reference information, which includes the pixel clock, the line clock, and the frame clock typically. So these are the three main components of the TTM. The TTM is an FPGA-based system. So we're using a Kintex 7 evaluation board from Xilinx. And the choice for an FPGA was based on several reasons. On the one hand, using an FPGA allows you to have a more versatile and flexible system because you can reprogram the circuit. So if we come up with a newer version of our software, we can very easily upload it to our board and also upload it to the cloud so that everyone else can use it. So it allows for a very flexible and versatile system. It's also a low-cost system. All the components combined will cost you about $3,000. So it's not that much for measuring arrival times of photons in more than 25 channels in parallel. And you have a temporal resolution in the order of tens of picoseconds. So it's more than good enough for typical fluorescence-based applications. So here on the right, you see a picture of the hardware. In red, you see the detector connector board. So this is where you connect your detector to the FPGA. The one on the left here was one of the first detector connector boards that we developed for 25 pixels. And so you had to physically connect every pixel of your detector to one of these connectors here. Then recently, we also moved towards detectors with 7 by 7 pixels. And then we implemented in such a way that you can simply connect the whole connector with all the pixels into this connector board. Then in blue, you see the board that is responsible for the data transfer to the computer. Right now, we typically use a USB type of connector, but we're also thinking of using the PCI Express in the future because that allows you for more efficient data transfer. And then here in green, you see these 4 connectors, which are used for the synchronization signals, so for the laser pulse, and also for the pixel, the line, and the frame clock. And then underneath the fan, that's the core of the TTM, the heart of the TTM, which is where the time tagging actually happens. I will not go into the details of how exactly it works, but since it's not unimportant, I will give, let's say, a brief overview of what happens when you want to do photon time tagging. So, like I said before, we're working with an FPGA, which runs with a frequency of 240 megahertz, which corresponds to a periodicity of 4.2 nanoseconds. And 4.2 nanoseconds is, of course, not good enough for doing photon time tagging because we want to measure the arrival times of the photons with picosecond range precision. So what we do is we have what is called these flash TDCs. TDC means time to digital converter. And it's basically some kind of chronometer. So what happens is when a photon is detected, then this chronometer is turned on. And you can think of this chronometer as a chain in which an electrical signal flows. And this signal flows until we observe the next clock cycle of the FPGA. So it runs for a certain amount of time, and it runs physically through this chain until it stops. And then you can simply check how far the signal traveled, and you can convert that to a time because it's directly proportional to the time. So then you know with about 30 picoseconds precision, the time between the photon and the next laser clock. You can do the same for the laser. So you wait until you observe the next laser sync. Then you start a second flash TDC, so a second chronometer, and the signal starts flowing again through this chronometer until you observe the next FPGA clock, and then it stops running again. And then you measure the distance that the signal has traveled through this TDC, and that is converted then into a time delta T. So these TDCs have a precision of about 30 picoseconds. So you can measure these 2 times with about 30 picoseconds precision. Then what you do is you check also how many clock cycles there were in between these 2 events. Then you do some basic math, and then you know with very high precision the time between the incoming photon and the next laser sync. And then when you know that, you can also find the time between the incoming photon and the previous laser sync. So then you have the information that you actually are interested in. I would like to show some of the key specs of the Bright-Eyes TTM. The single shot precision of the device is about 30.3 picoseconds. Single shot precision means that if you feed, let's say, a photon signal into the TTM and also a reference signal with a fixed delay, and then you measure the delay that you get with the TTM, then you can make a histogram like this one. So what we did was we fed a photon signal to the TTM and then a reference signal about 18 nanoseconds later. And we registered this delay with the TTM. And you can see that you get a histogram with a very sharp peak around 18 or 18.5 nanoseconds with a standard deviation of about 30 picoseconds. So you can say the single shot precision of the device is around 30 picoseconds, which is very good because it's below the jitter that the detector itself has. So the TTM performs better than a detector, which is of course what you want because you don't want the TTM to be the limiting factor. Then we measured the impulse response function of our system. Impulse response means that if you place a sample on a stage that doesn't have a lifetime, so that responds immediately, then ideally you also want to have, you want to see this immediate response in your signal. But because of various reasons, because of various timing jitters, this is not literally an infinitely small peak. But instead you get a response that has a full width half max in the order of about 200 picoseconds. You can see here in blue the comparison between the Bright-Eyes TTM and in purple the Becker and Hickel system, so a commercial system which has a full width half max of 250 picoseconds. So our device performs slightly better than the commercial system. So again, what you see here, this impulse response is the response of the whole system, not only of the TTM itself, but also of all the optics involved, all the electronics involved, and the detector in the system. But again, 200 picoseconds or 250 picoseconds is well below the typical lifetime of your sample. So it's perfectly suited for measuring fluorescence lifetimes. Then the sustained count rate is around 125 megahertz. Sustained count rate means the maximum photon count rate that the system can handle before it starts saturating. So in order to measure this, we fed a certain photon count rate into the system and we checked what the system measured. So the photon count rate that the system measured. And you see in green that it is linear up to about 125 megahertz, and then it starts saturating. So it means that you cannot detect more than 125 million counts per second over all the channels combined. But again, this is typically much higher than the photon count rates that you actually have in a typical microscopy setup. And then we did some measurements of the fluorescence lifetime to see that the system performs properly. Just a brief recap of what fluorescence lifetime actually is. So in fluorescent molecule, you have the ground state, and then a laser beam can be used to excite the molecule to an excited state where it stays for a certain amount of time, typically 3, 4, 5 nanoseconds. And after this time, it decays back to the ground state. And this time is a characteristic time for a molecule, but it also depends on the environment in which the molecule is. So measuring this lifetime gives you information on the environment in which the molecule is, and so on the sample. So what you typically do is you measure a histogram of the photon arrival times, you fit an exponential decay through it, and then this tau value gives you the characteristic lifetime, which, like I said before, depends on the molecular environment or on the pH or conformational changes and so on. So this is a very interesting property to measure, and we did some test measurements on a sample of fluorescence with different concentrations of quenching material, which in this case is potassium iodide. And so when you have pure fluorescence, which is the curve you see in blue, then you get a relatively long decay time. And then if you increase the concentration of the quencher, you decrease the lifetime of the molecule and you get a faster decay. And this is just to show that we can measure these differences with the TTM, and we can do this for 25 or even 49 channels simultaneously. Another key advantage of our TTM is that it can be used with any laser scanning microscope because it works as a slave system and it's a plug and play device. So you can use it on a custom built microscope, but also on a commercial microscope. For example, we tested it on the Nikon A1 confocal microscope, which doesn't have a spat array detector, but a PMT. But even in this case, you can use a TTM for measuring fluorescence lifetimes. So I think this is a very important advantage and an important feature to highlight. Already mentioned before, it's customizable. So that's one of the reasons why we use FPGAs. And so if you have different needs, if you have a different number of pixels, or if you have a different different type of measurement, then you can change the code according to your needs. Right now, in the current implementation, we use only about 20% of the FPGA resources. So just to show that there is plenty of room left for more channels or for more complex analysis, let's say. And then, like I said before, it's a very low-cost system. It will cost you about $3,000 to implement this system. So this is also a crucial advantage compared to many of the commercial solutions. Okay, with this, I have highlighted most of the technical aspects of the system. Of course, I'm open to questions, but right now I would first like to give the floor to Eleonora, who will highlight some of the important applications that you can do with this Bright-Eyes TTM.
So yeah, thanks, Eli, for introducing the Bright-Eyes TTM. I will go through some of the application where we actually use the Bright-Eyes TTM into our system. That would be mostly with spot array detector, but yeah, as Eli showed, we also tested our time tagging module with several other microscopes. So it's really fully compatible with other systems. So what can we do with the Bright-Eyes TTM and the spot array detector? So with the Bright-Eyes TTM, as Eli was saying, we really can tag each single photon arriving on the detector in time. And thanks to the spot array detector, we also have spatial information. So we are having like a bunch of single photon measurements where for each photon we know exactly the time and the space it arrives. I will start show you what we do, starting from imaging in the context of imaging, how we can, for example, implement the fluorescence lifetime, especially in image scanning microscopy. So here you see basically all the temporal range we can access and the space range we can access. You see that we can have we have a wide range, both in time and in space. So again, a small reminder on fluorescence lifetime, we have basically the Bright-Eyes TTM that tag in time each single photon that arrives. With the arrival time, we build a so-called fluorescence arrival time histogram or start-stop time histogram, where we can fit with a single or multiple exponential function, and we get the lifetime of this fluorescent molecule, which is simply the average time the molecule spent in the excited state. And I'm saying single or multiple exponential function because this can actually vary. One sample can have either one single exponential decay, so one lifetime, or it can have multiple lifetimes, depending on, for example, the environment where it is. We can combine it with imaging by thinking that for each pixel we are acquiring, so for each position of the laser, we can actually build one of these histograms. And then we have, what we can do is basically two approaches. One is this one, where we fit each single pixel with an exponential function. And for each pixel, we get a tau value, so a lifetime value. And then we can create so-called fluorescence lifetime image. So in this case, each pixel is not reporting the intensity, but it's reporting the value of its lifetime. And this is recently starting to really increase the curiosity of scientists, because it can bring your imaging from structural imaging to functional imaging, reporting, for example, changes in pH, changes in ions. How can we actually insert it in FLISM? We can combine it with image scanning microscopy, passing so from FLIM to FLISM. And in this case, for each bin of the histogram we have, we reconstruct the ISM image. So for each time bin of the histogram, of the start and stop histogram, we have now an ISM image. And then we get a super resolved image for each time bin. So we can simply connect the two information and create both a super resolved image with the information of the lifetime. And this is the first approach. The second approach is more visual. It's called phasor approach. Basically, for each histogram, what we are going to do is transform the histogram into two coordinates through the Fourier domain. And then we plot these coordinates into real and imaginary complex. Basically, it's a complex number. We can plot them, and they will lie in a so-called universal circle. The universal circle represents all the values of lifetime. On the right, we have short values of fluorescence lifetime. On the left, we have long values of fluorescence lifetime. And you can calibrate the universal circle on your system with a fluorophore, you know the fluorescence lifetime, because it depends on the system, and it depends, for example, on the frequency of the laser pulse you use. So because one really cool effect is that because the histogram of the photon arrival can be described as an exponential into the phasor space, two different lifetimes just linearly combine if you have a mixer. So if you have a single value of lifetime, it will lie on the circle, on the universal semicircle. If you have a mixer, so a multi-exponential case, the value will lie inside the circle. So if you have fluorescence lifetime, that's combinations of different values, they will be inside the circle, and you can actually decompose them with algorithm to go back to the each single component fluorescence lifetime. So for testing this out, we start imaging fluorescent bits. Here you see, again, the three types of microscopy that we can do that Eli also explained before. We have the confocal with a very close pinhole, and on the top left, you see intensity-based images. On the bottom right, you see fluorescence lifetime-based images. And as you can see, in the confocal, we have actually a really good space resolution, but the level of noise is quite high. And this is reflected on the histogram of the lifetime we are measuring, and also on the phasor plot. You see how this cloud is broad. If we actually consider the whole area of the spada ray detector, so performing conventional confocal microscopy, we have a really we have improved the signal to noise ratio, but the space resolution is lower than before. And you see how we have still a quite broad cloud of points in the phasor plot, but it's very restricted compared to before. In the ISM reconstructed data, we have the best of both worlds. So we have high intensity and a good spatial resolution, and this is also reflected in the phasor plot. A more biological application to test the functional imaging, we use it on live cell. These are X cells stained with dye that's called ANEP. This dye is incorporated into membranes, and interestingly, its lifetime changes depending on the polarization of the lipid it is inserted. So you see here in the FLISM image, you actually are able to see two different lifetimes. We can even do better just by creating the phasor plot. So each decay is plotted into the phasor system. You see that the cloud is inside the universal circle, hinting on the fact that we have more than one component of lifetime here. So what we did was simply segmentate the pixel of the image based on the phasor plot. We have a line passing through the center. We divide the short lifetime from the long lifetime, and this time we plot the intensity-based image, but with two colors depending on the two lifetime. And you see that we are able to distinguish two types, basically, of membranes inside the system. But with this, we can also perform more. We can, I show you how we can use the temporal tags to perform lifetime combined with imaging, but we can also perform spectroscopy measurements. So, for example, fluorescent correlation spectroscopy or lifetime correlation spectroscopy and cross correlation. And today I will go a bit more in detail in the first two. First, what is fluorescent correlation spectroscopy? Quickly, basically, it's not a scanning method anymore, but we are fixed into one point. It was developed around in the 70s to measure the movement of fluorescent probes. What happened is that we are acquiring over time from one single point the intensity fluctuation caused by the movement of the particle inside out. And with this, what we have, it's basically this. We have the fluorescent fluctuation over time, which will look like this. And these peaks are caused by entering of our molecule. What is done typically in fluorescent correlation spectroscopy, or FCS, is autocorrelating the signal with itself on different lag time. There are so-called this tau value. And this will give you a measurement on how long a molecule is inside your volume of observation. Knowing the size of the volume of observation, we can actually derive physical values on dynamics. For example, the diffusion coefficient or the concentration. And depending on the diffusion coefficient, as you see here, we have different decay curves. So for a bigger molecule, which will have a slower diffusion coefficient, the decay moves towards the right. For smaller molecule with a higher diffusion coefficient, the decay moves towards the left. We are able also to distinguish the concentration and to measure the concentration of your sample, because it is the number of molecules in the volume. It's inversely proportional to the amplitude of the FCS decay curve, as well as multiple components in the system, which will appear as two different decay peaks. And this is conventional fluorescent correlation spectroscopy. However, we are doing it with SPAD array detector. So what we can do is actually have 25 channels, not only one. And what we are having here, because the SPAD array detector is basically as a small camera with infinite frame rate, we are basically directly imaging the detector volume. So one trick we used was to perform so-called spot variation FCS, trying to make the most of the spatial information of the SPAD array detector. Here, for example, you have molecules that are diffusing freely outside, but they are confined also into a condensate. And with conventional FCS, you will not be able to distinguish the two types of movement. What we can do here is actually going to probe different position, different areas and volumes of your samples to actually distinguish, being able to distinguish these different movements. And we can do it by simply a post-processing phase. What we do is integrating the signal coming from different positions, from different channels of the SPAD array detector. And with that, we can create with one single measurement. We have three actually measurement of three volumes in your sample. With that, we can perform the so-called diffusion law, where plotting the diffusion time decays for a different area of the detector gives you an idea on how the molecules are moving. So we know not only the velocity, but also the type of movement, if it's free diffusion, if it's moving into a domain confined system, like, for example, forming condensate, or if it's diffusing into a dense meshwork, it will have a different a different intercept of the linear regression. And with this, we can directly investigate different volumes in the sample. One example, we did performing measurement into a living cell expressed DEC protein. DEC protein is a protein that is interacting with the chromatin. What happens if that we do this measurement in the cytoplasm with DEC is not interacting with anything, we are able to retrieve its value of diffusion, and also the spot variation FCS measurements tell us that's freely diffusing in the sample. However, if we perform this measurement inside the nuclei, where that is interacting with the chromatin, we see a completely different decay function, and the intercept is bigger than one. Hinting towards a confined diffusion, or in any case, some sort of interaction behavior of this protein within the system. Another example on how we did implement the lifetime information, in this case, with this sort of measurement, it's here where we use a technique called circular scanning. So now, instead of fixing into one point, we are also scanning the sample. And this gives you the formation of this oscillation in the system. What we did is performing something similar as before. We integrate in a post-processing phase the signal coming from the three different volumes, and we get the three curves. However, what we noticed was that the number of molecules inside the volumes were not consistent with what we expected. And this was because we had some artifacts coming from the detector. That's why we use the fluorescence lifetime. So we know that this, in this case, beats our sample was decaying with a lifetime of about 2 nanoseconds. So what we are going to measure into this histogram is that only the photons coming from the first, let's say, nanosecond are coming mostly from fluorescence from the sample. However, all the photons coming here are most likely due to the detector artifacts. So what we did was implementing a filter directly based on the fluorescence lifetime to filter out all the photons coming from this region. And you see here, these are the filtered autocorrelation curves. They look more noisy because we have less photon, but this time we are able to recover a number of molecules in the system that's in agreement with what we were expecting, as you can see here. Another application we did, combining the two sides of the story, is checking how GFP is diffusing into a cell. Here you see intensity image-based, fluorescence lifetime image-based, and what we can do also is choose a point and do a measurement. And in this position, we did both the FCS measurement, measuring then how it moves, but also the fluorescence lifetime measurement. We are here, it could give us information on possible changes on the in the function and in the structure at the same time. So we can really follow the temporal evolution of the system in all its value. And yeah. Another application where this comes really to a big importance is studying, for example, the so-called liquid-liquid phase separation in cells, where organelles are formed. In this case, I'm showing you two cells, how they react with stress. Upon stress induction, we see the formation of the so-called stress granules, which you see here. The stress granules are just condensate of proteins and RNAs, which are used from the cell to sort of segregate the important molecules during the stress. When the stress is gone, they will be released back into the system. So they are called liquid organelles, in the sense they are membraneless organelles, and they can be basically dissolved once the condition of stress is gone. And these are important for cells. This is just one example where it happens, but there are several other processes where this liquid-liquid phase separation takes place, and it's used from the cells to actually organize in space and time different processes. By conventional microscopy, what we can see is just the, okay, before the stress, this protein, which is G3BP1, it's a reporter of stress. It's freely let's say uniformly in the cytoplasm of cells. Once we have stress, we have the formation of stress granules. What we can do more is, for example, combining lifetime. Here you see again intensity, fluorescence lifetime imaging upon stress. What we did was perform a phasor plot analysis, and we again are able to distinguish actually the short lifetime from the long lifetime. And in just by lifetime, with no fitting with the phasor plot, we are able to automatically segmentate the stress granules inside the cell. We can also combine it with let's say dynamic measurement done with FCS. Before the stress, we see that the G3BP1 is moving mostly freely inside the cells, while after the stress, we measure an increase of this intercept, which is related to formation of condensate. And we can finally mix all the information we have together to actually follow both, let's say, the diffusion coefficient with the lifetime and imaging. And this tells us, for example, that once the formation of the stress granules started, the diffusion coefficient decreased, so we have a slowdown in the motion of G3BP1, and simultaneously the lifetime decrease, which hint for a fret-like process or self-quenching. So there's some transfer of energy inside these stress granules. And this is a way we use to actually connect, let's say a smart way to use the so-called single-photon microscopy, allowing us to connect different processes on different time scale and space scale, in this case, related to biology. So to conclude, we show you the BrightEyes TTM, which is a low-cost open-source time-tagging molecule for single photon microscopy. We show you how we can use it on multi-channel parallelly with high temporal precision. It is a plug-in and play design, and you can use it on any custom build or also commercial setup. And I show you how in combination, the combination of the BrightEyes TTM and the sputter ray detector allow us to unlocking multidimensional space and time measurement. And with this, we hope we can bridge structure and function in cells. One last slide. So this is part of, let's say, an open science mission of our lab. We don't have only the BrightEyes TTM as an open source project, but the idea is to really have so-called BrightEyes scope, instruction where you can assembly by yourself low-cost single-photon light scanning microscope, and we have also our own open source microscope control. On our GitHub page, you can find how to build microscope, how to build the BrightEyes TTM, and also how to analyze data and some data. This is our group, which we want to thank everyone in and all of our collaborators. And just a small sponsor, we are looking for both postdocs and PhDs. So if you want to travel and live in Italy for, just try out, you can either do a PhD with us. We are looking for people more expert in optics and hardware development, but also postdocs more expert into life science. And with this, we are open for questions.
Yep.
Thank you very much. That's really fascinating. I'm really amazed what you can do with that. And also, I think you mentioned a couple of measurement methods that I actually didn't hear about before. So it's pretty cool, the kind of things you can do with your device.
Thank you.
Yeah, I do have a number of questions. I don't really know where to start because there were so many interesting things there. Maybe to go to the technical part first. I do have some questions about the timing. So you were talking about different methods and different applications, but all of them were working a little bit on different time schedules. And especially when you were talking about the TTM, if I understand that correctly, then you said that it takes quite long to wait for individual photons. And I guess you have to, if you because you did combine that with fluorescence lifetime measurements, right? So then I guess you need to collect a number of those on each position before you can calculate a good value. So then I was wondering, how fast can you actually scan through a microscope image with that? So I guess how long do you need to measure at each point to get a good
quite
value?
fast to have a good measurement. It's enough to have, let's say, 50 microseconds per pixel, but even way slower. It really depends also on your sample.
Yeah, the limitation is not in this case. Yeah, not the TTM, but indeed the sample. So we wait for a photon to arrive because typically a laser has a pulse frequency of 40 or 80 megahertz, and the photons coming from the fluorescence are typically not with this frequency, but maybe 1-2 megahertz, something like that. And so it's more efficient to wait for a photon and then measure the laser pulse rather than the other way around. But this is also true for let's say normal confocal microscopy with a single channel. So the frequency of the emission is in the order of a couple of megahertz, while the frequency of the pulses is maybe 80 megahertz.
Okay. I see, but it's still pretty fast overall.
Yeah.
That's really nice. And then on the last now, I don't remember exactly how to call it, but Eleonora, you were talking about this application where you could see how do molecules move and diffuse in the sample. I think that's really fascinating.
Yeah.
But there you don't typically scan an image, right? So that's only on one point of the sample where you can do this measurement. How long do you go there?
Yeah, so this is, let's say, the basic usage of this method. There are also methods where you actually scan the system and then you have a value of this movement for every pixel, but it is valid only for very slow moving molecule, let's say. And also there, it's enough to collect a good signal to noise ratio, let's say a good amount of data, which is depending on the signal to noise ratio. So for example, a sample of bits, it's enough a couple of seconds to have a good value.
So typically when we do a measurement in cells, for example, then we first take an image. So we scan the sample to see where the cells are. And then we choose a position where we want to do FCS, so the cytoplasm or the nucleus. And then we park the laser beam there for several tens of seconds typically. So we park the beam then in a fixed spot for the measurement.
And then you switch the acquisition mode of your system into the other measurement kind.
Exactly, yeah.
That's great. Yeah, well, it's fantastic if you can do that with the same system. I guess it's quite easy to switch.
Yeah.
That's really cool. I was also very fascinated with this time tagging principle that you described. It took me a while to understand, but that's really incredible how you managed to increase the time precision of the system much beyond the clock of the FPGA. And I was wondering, have you heard of the White Rabbit system that was developed by CERN so they use that for time clocking, also particle decomposition events across kilometers of instrument, because they have the large particle accelerator and they synchronize the measurements. But I don't actually know how it works and if it has any similarity.
I read about it today, but I don't know either. It's quite interesting.
Okay.
No?
Yeah, no, me neither. No.
Because that's also open source hardware. So I guess, yeah, I mean, there are other commercial systems, but they will not tell you how they do it. So in the open systems, I just had that thought that there was something related. Yeah, that's really, really cool. But I guess it's difficult to apply this to systems where you have a higher photon flux, right? So this would only work for rare events, right? Because you can only track the time of a single event within a clock cycle. So if you have several events, you don't know?
No, we can. Yeah, so the photon count rate is limited to about, like I said, 125 megahertz. So for most applications is enough, because also the spot Yeah, in fluorescence at least, because the spot array detector also has a certain dead time. So when a pixel receives a photon, the pixel is blind in any case for a certain number of nanoseconds. So yeah, then the DTM doesn't need to be to have a much higher ability than what the detector itself can do.
Right. Yeah, so that's actually really convenient in that case, the blind time then for your for your
Yeah.
timing. Okay, that's really cool. Well, we're thinking about we're planning on building a kind of Raman spectroscopy system for high throughput measurements, and a lot of the technical issues that you're explaining here are also surfacing there in a different way though, but we always have a relatively high signal. So
and what is the order of magnitude of the signal that you have?
I'm not sure in these time frames to express it.
Okay.
Dig into it, and I have to see
Yeah.
run the numbers. I haven't done the math.
Yeah. Okay.
And well, there are also different measurement modes. So I guess there's the sort of coherent Raman. Maybe that would work with this kind of system. The stimulated Raman that we would like to do that always has a high base value, and you try to look at fluctuations of laser intensity. So then maybe it's a bit more tricky.
But in any case, this Bright-Eyes TTM is also still under development, so it's possible that in the near future this limit will increase. So we're continuously working on this.
That's really impressive. And the slide before, the slide 13 that you had, can you give us another idea on what of these parts did you really assemble from scratch? And so, for example, this Xilinx card, I mean, Xilinx, of course, is just the FPGA chip, but this whole board, did you solder all the components, or that entire card, you buy it and you develop just those kind of breakout boards to interface to it, or how?
Yeah, exactly, like that. So we didn't do much of the hardware ourselves, let's say. So you can buy most of these components and then you assemble it. And then most of the work is on the software side. So on coding everything on the FPGA level, and then also for the data processing. Yeah.
Okay, so the data transfer card, including that's also something that's commercially available.
Yeah.
Yeah. Well, I guess it's helpful. So if you have to, because they're quite big, complex boards. So if you have to solder everything, it's a lot of purchasing and soldering.
No, I think you don't need to be an expert in these things in order to build the TTM. So the instructions are online, and I think if you have basic knowledge, it's more than enough to build your own device.
Yeah, congratulations also for working with FPGA. So of course, in this context, it's hard to get around it. But I found that, so I started looking into working with FPGAs for obvious advantages a few years ago. And now we have a project also that was implemented with FPGA, but it was surprisingly difficult. Like it's on a very different technical level to code the FPGA and also upload everything onto the FPGA. So I'm wondering what your experience there was. Yeah, in the end, we work with the Bright-Eyes board, which can actually be programmed mainly in Python, even though it still works with FPGA to make it like a little bit more biology developer friendly. But it looks like a pretty complex job that you managed to do here.
Yeah, most of the work was actually done by two colleagues, Mattia and Alessandro Rosetta, regarding then the coding of the FPGA. But yeah, it's true. It's very difficult, very complex also to debug it, because it's not so straightforward compared to high-level coding. But yeah, in the end, we managed to do first for 25 channels, and then later we upgraded it to 49 or even 50 channels right now. So yeah, that's
really impressive. So obviously, I'm completely sold on the idea of open source hardware. But I'm wondering to hear a little bit from you. What was the motivation for you to release this as open source hardware? Do you know, have you been part of that discussion from the beginning, or was just like a lab policy already, or what were your thoughts on this?
Yeah, the policy of our lab is already from a long time to have this open science ID. So also in another project, we upload our code and our data and everything. And then, yeah, when we wanted to do this time tagging, of course, you check first what is already commercially available, but then when you look at the price and the number of channels that you need, then it's very difficult to find something suitable. So then it came to our minds to build our own TTM. And since we have this open science philosophy, we want to make this open source also.
Fantastic. Well, thank you very much for leading us a little bit into the details of both the technology that you developed and how it works, and also what it can be applied to. So yeah, that sounds like there's a lot of promise still for exploring many different biological questions with this. So thank you very much once more.
Thank you for the invitation.
Thank you. Yeah.
Well, thanks for coming. Yeah, and thanks for being flexible in time. Now we overran a little bit, but it was great that we could still discuss.
Yeah.
All
Thank
right.
you.
Are there any other questions? If not, we'll just close the seminar. So yeah, thank you. And I hope to meet you someday in some bioimaging or hardware development context on some conferences.
That would be great.
Yeah.
Yeah.
Nice to meet you online.
Yeah.
Bye.
Thank
Bye.
you.
Thank you.
Bye.